Right, so we're going to have a look at this question, which is from summer 2013 for B2, and it's the question you've been stuck on about the Hexa gene. And it tells us that a gene codes for the production of an enzyme called Hexa. So as you know, enzymes are made out of protein, and proteins are coded for by genes. And this is about genetic disorders. So there's a genetic disorder that damages the nerve cells in the brain. And that disorder is caused by a small change in the DNA of the Hexa gene. So basically, you've got a faulty version of the Hexa gene, which codes for an enzyme that doesn't work, and you've got a version of the gene, the normal one, which does work. So people with the disorder make a changed Hexa enzyme that doesn't work, which is what I've just said, basically. So the question, it's an explain one. Alright, so we've got to go into some detail here and say why it is that a change, so explain goes with the word why. So why is it that when you change the DNA of the Hexa gene, that an enzyme is produced, a Hexa enzyme is produced that does not work, does not do its job properly. Now I gave Sakib 3 out of 3 for this, but before we have a look at what he's put, let's have a little look at the science behind this. Now I think it's worth having a look at this because in the exam spec it isn't that clear that you need to know a bit about this. Now you don't need to know everything I'm going to go through now, but I think a bit of understanding on this is very helpful in terms of answering the question. Here's our DNA, double helix structure, and the bit that does the coding for DNA. Now in the exam spec it doesn't use this word, but it's these coloured things here, and the word is bases. These are bases. There's 4 different bases, and every 3 bases codes for 1 amino acid. Now what you most definitely need to know for the exam is that this DNA code determines the sequence of these amino acids, and amino acids make the protein, so they're the building blocks for the protein. And of course enzymes are made out of protein. So this is the key science for the question. Now let's go back to Sakib's answer and we'll have a little look at the mark scheme as well. So Sakib has said that a change in the DNA code for the change in DNA will code for a different amino acid sequence. I've still given him 3 out of 3, but I think he should have been clearer. He got a bit confused with that term base that I mentioned. So a change in the DNA code will mean that we're coding for a different sequence of amino acids. And then the amino acids are used to produce the enzyme, which is of course made out of protein. So different amino acids mean a different structure for the enzyme. And that means the active site on the enzyme will be changed, so the substrate will not be able to fit in, and therefore the reaction won't be catalyzed, the enzyme's not able to work and do its job properly. Let's look at the mark scheme. You can play this back and pause things if you like. So I've given him 3 out of 3 because he's got 1 mark for referring, well he's actually done both of these, he's referred to a change in the code and changing the sequence of the amino acids. He's referred to the structure of the enzyme or the protein being changed. He's also referred to the active site too. And he's done the last bit too, talking about how the substrate will then not fit into the enzyme and therefore the enzyme won't work. Let's have a little look at the rest of this question. Fairly long question, so this clip's about going to be slightly over 5 minutes, I reckon. How many chromosomes are there in the nucleus human nerve cell? You need to know it's 46. 23 pairs, which makes 46. Jobs are good. Let's move that away. We're finished with that now. Next one. So, the boy had a changed Hexa gene on his chromosome number 15 that he got from his father. But the changed Hexa gene coded for a Hexa enzyme. So we know that the changed Hexa gene codes for a Hexa enzyme that does not work. But the boy did not develop the genetic disorder. Explain why he didn't. Okay, so basically, as Sakib's sussed out here, the Hexa gene must be a recessive allele. Okay, it must be passed on by a recessive allele. So you need 2 copies of that recessive allele, one from both parents, to inherit the disorder. So that's for 1 mark. You don't need all of what I said, we'll see the mark scheme in a sec. But what you do need is this key thing, that he got the normal gene, which is the dominant one, from his mum. Alright, have a little look at the mark scheme. Here it is. That's the 46 from before. So 2 marks, 1, 2. He inherited from his mum a normal allele, or you could call it a normal gene, or you could refer to the fact that the boy must be heterozygous, he's HH, which I added to my notes. Or, like Sakib said, and, not or, for the second mark, the allele must be recessive. Okay, or you can say the normal allele would cause the enzyme to work, because that, of course, is the dominant one. Last little bit of the question, here it is. So this boy, who we know is big H, little h, with big H being the normal, little h being the recessive one for having the genetic disorder. So he's got married. So we're making sense of this as we go through. We're not rushing straight on. The blood test showed that his wife also inherited that dodgy gene. And it tells us there's a 1 in 4 chance. Alright, so that is a big clue there. That tells us from the genetic crosses we've done before, we know that if both parents are heterozygous, there's a 1 in 4 chance of the inheritance of, either 2 copies being homozygous for the dominant one, or in this case what we're focusing on, 2 copies of the recessive one. So we've got to explain why that is. Why is there a 1 in 4 chance, so I've not actually said this last bit, that the couple's first child will have the genetic disorder? So you've got to explain it. So for your 3 marks, you need to clearly show that both parents are heterozygous, they've got 1 of each. You then do your genetic cross, you get the 2nd mark for that, and then you get the 3rd mark for highlighting the 25%. Easy peasy, you can do that. There we go, like we just said, 1 mark for labelling both parents being heterozygous, 1 mark for correctly working out those outcomes, those what we call genotypes of those possible genotypes of the offspring, for deriving those genotypes, and thirdly for identifying the HH being 1 out of 4. Excellent, that's it. That took a while, but that's everything you need to know about that question. See you later.